Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Agriculture, SOA, Master's Degree Programs, Master of Science in Food Safety and Quality Management, MSCFSQM, First Year, MVPI 001 Food Microbiology, Block-1 Fundamentals of Food Microbiology, Unit-3 Food-borne Diseases, 3.0 Objectives. After reading this unit, we shall be able to specify foodborne diseases, their types and causative agents, enumerate common foodborne diseases and their symptoms, and state emerging foodborne pathogens and reasons for their emergence. 3.1 Introduction. Foodborne disease is caused by consuming contaminated foods or beverages. Many different disease-causing microbes or pathogens can contaminate foods, so there are many different foodborne infections. In addition, poisonous, chemicals, or other harmful substances can cause foodborne diseases if they are present in food. More than 250 different foodborne diseases have been described. Most of these diseases are infections caused by a variety of bacteria, viruses, and parasites that can be foodborne. Other diseases are poisonings caused by harmful toxins or chemicals that have been produced in or have contaminated the food. For example, mercury in fish, seafoods, mycotoxins in peanuts, wheat, etc. These different diseases have many different symptoms, so there is no one syndrome. That is foodborne illness. However, the microbe or toxin enters the body through the gastrointestinal tract and often causes the first symptoms there. Nausea, vomiting, abdominal cramps, and diarrhea are common symptoms in many foodborne diseases. Many microbes can spread in more than one way, so we cannot always know that a disease is foodborne. The distinction matters because public health authorities need to know how a particular disease is spreading to take the appropriate steps to stop it. For example, Escherichia coli O157 H7 infections can spread through contaminated food, contaminated drinking water, contaminated swimming water, and from toddler to toddler at a daycare center. Depending on which means of spread caused a case, the measures to stop other cases from occurring could range from removing contaminated food from stores, chlorinating a swimming pool, or closing a child daycare center. 3.2 What is disease? The term disease is applied to any harmful change in the tissues and/or metabolism of a plant, animal, or human that produces the symptoms of illness. Microorganisms, bacteria, yeasts, molds, viruses, and protozoa that cause diseases are known as pathogens. 3.3 How do microorganisms cause disease? Soon after birth, the external surfaces and cavities of our bodies are colonized by large numbers of different types of microorganisms that originate from other humans and the environment in general. These organisms constitute our natural, permanent, native microflora. Most of the organisms are bacteria, but some yeasts also occur. This natural resident microflora is symbiotic, i.e., it lives in mutual harmony with our body tissues and is essential for our well-being. Here are two examples of importance of our permanent microflora. Times the permanent microflora is essential in combating invasion of the body by potential pathogens by competing for space and nutrients, and sometimes producing antibiotics. The presence of a variety of strains of Escherichia coli in the colon, for example, helps to prevent enteric pathogens such as Salmonella spp from becoming established. Laboratory animals that are reared under sterile conditions and without a natural resident microflora are exceptionally prone to diseases caused by organisms that are not even normally considered pathogens. Times bacteria in the colon synthesize vitamin K and contribute significantly to our requirement for this vitamin. Our bodies are constantly being exposed and infected with organisms that are not part of this permanent microflora. Most of these organisms are harmless and transient. Others are pathogens and have the ability to invade are tissues or produce toxins, or both. Toxins are chemical substances produced by microorganisms that are harmful to human tissues and physiology. Many, but not all, of the toxins produced by microorganisms are proteins. Sometimes toxins are secreted into the environment in which the microorganism is growing. For example, the enterotoxin produced by Staphylococcus aureus can be secreted into food. Toxins of this type can come into contact with or enter the human body and cause disease in the absence of the organism. 3.4 Foodborne diseases and the agents. Foodborne disease is simply disease that results from the ingestion of food. Foodborne diseases, FBD, are defined by the World Health Organization as diseases of infectious or toxic nature caused by, or thought to be caused by, the consumption of food or water. More than 250 FBDs have been described. Symptoms vary widely, depending on the etiological agents. Diarrhea and vomiting are the most common. In many countries, national healthcare organizations record FBD outbreaks, defined as the occurrence of 2 or more cases of a similar illness resulting from the ingestion of a common food. Agents that can be responsible for foodborne disease are 1. Microorganisms, 2. Parasites, 3. Chemicals, 4. Naturally occurring plant toxicants, 5. Naturally occurring fish toxicants, 6. Metabolic disorders, 7. Foods that give rise to allergies, 8. Radioactive materials. Common food poisoning agents are given in the table. 3.1. Microorganisms are by far the most important agents of foodborne disease, with bacteria causing the major bulk of foodborne disease outbreaks. Viruses are also an important source of foodborne disease, with foodborne transmission of infective protozoa far less common, particularly in developed countries. Some molds produce substances that are toxic to man, mycotoxins, but their importance in foodborne disease production is currently not known. A few algae produce toxins that are associated with shellfish poisoning. Yeasts are very rarely associated with foodborne disease, apart from the ability of Saccharomyces cerevisiae to produce alcohol. The one documented example is associated with a yeast that infects the surface of sun-dried fish in South America. The organism can cause a skin infection in anyone handling the fish. Prions that cause degenerative diseases of the nervous system, e.g. the agent causing bovine spongiform encephalopathy, BSE, in cattle, may possibly be transmitted to man via infected offal. Table 3.1. Common food poisoning agents. Agent example: bacteria Salmonella, Staphylococcus aureus, Clostridium perfringens, C. botulinum, Bacillus cereus, viruses hepatitis A virus, parvovirus, Norwalk virus, moles Claviceps purpurae, Aspergillus flavus, foodborne, aflatoxins, marine protozoa. Gonulax tamieusis, paralytic crustacean, poisoning. Gambiodiscus toxicus, poisoning with Ciguatera. Prorocentrum lima and some species of Dinophes, direct shellfish poisoning, parasites, protozoa, amoebas, Trichinella spiralis, Taenia solium, Giardia lamblia, Cryptosporidium, Entomoeba histolytica, chemical substances, heavy metals, lead, zinc, copper, cadmium, mercury, arsenic, antimony, pesticides, herbicides, fungicides, substances used for cleaning and disinfections, toxic plants, germinated potatoes, apricot core, red bean, Phaseolus vulgaris, toxic animals, combrotoxin from fish insufficiently cooked. Herring, histamine. 3.4.1 Diseases by bacteria. Toxins are particularly important in production of bacterial diseases. Bacterial toxins are classified into 2 types: exotoxins and endotoxins. Exotoxins have the following characteristics: Z. Generally proteins synthesized by metabolic activity. Z. Not structural components of the cell. Z. Secreted into the cell environment. Z. Produced by gram-positive and gram-negative organisms. Endotoxins have the following characteristics: Z lipopolysaccharides, Z toxic components of the cell wall released when the cell dies and breaks down, Z produced by gram-negative organisms. 3.4.2 Diseases by moles. Mushroom poisoning is caused by the consumption of raw or cooked fruiting bodies, mushrooms, toadstools, of a number of species of higher fungi, Basidiomycetes. The term toadstool, from the German to destil, death stool, is commonly given to poisonous mushrooms, but for individuals who are not experts in mushroom identification, there are generally no easily recognizable differences between poisonous and non-poisonous species. The toxins involved in mushroom poisoning are produced naturally by the fungi themselves, and each individual specimen of a toxic species should be considered equally poisonous. Mycotoxins are low molecular weight natural products, i.e., small molecules, produced as secondary metabolites by filamentous fungi. These metabolites constitute a toxigenically and chemically heterogeneous assemblage that is grouped together only because the members can cause disease and death in human beings and other vertebrates. Not surprisingly, many mycotoxins display overlapping toxicities to invertebrates, plants, and microorganisms. Unfortunately, mycotoxins can also be incredibly toxic to humans, causing a variety of responses including cold flu-like symptoms, sore throats, headaches, nosebleeds, fatigue, diarrhea, dermatitis, and immune suppression. Some mycotoxins may also be carcinogenic and teratogenic. Moles that have been known to potentially produce toxins. Some of these moles are Acremonium, Alternaria, Aspergillus, Chaetomium, Cladosporium, Fusarium, Penicillium, and Stachybotrys. Even though these moles may potentially produce mycotoxins, they will not do so unless specific environmental conditions exist. Common types of mycotoxins times aflatoxin. This mycotoxin is primarily produced by Aspergillus species. It is one of the most potent carcinogens known to man and has been linked to a wide array of human health problems times ochratoxin. This mycotoxin is primarily produced by species of Penicillium and Aspergillus. It can be damaging to the kidney, liver, and it is a suspected carcinogen. There is also evidence supporting its role in impairing immune system function times trichothecene. This toxin is produced by Stachybotrys spp and Fusarium spp and has even been indicated as a potential agent for use as a biological weapon. One of the more deadly mycotoxins. If it is ingested in large amounts, it can severely damage the entire digestive tract and cause rapid death due to internal hemorrhaging. 3.4.3 Diseases by viruses. Unlike bacteria, viruses invade host cells, take over host cell metabolism, and induce the cell to produce new virus particles. Disease symptoms are caused by the destruction of host cells and secondary effects resulting from host cell destruction. Several viruses like hepatitis A, Norwalk and Norwalk-like viruses, poliovirus, and echovirus may cause food-borne disease in consumers of virus-contaminated water and foods. Some of the other viruses that have also been associated with food are astrovirus, calcivirus, enteric adenovirus, parvovirus, and rotavirus. These enteric viruses replicate in the intestine of infected individuals and are transmitted by fecal-oral route. They must survive the acidic environment of stomach, the alkaline conditions and digestive enzymes of the small intestine, and the conditions encountered between the hosts. Such selective pressures result in a stability that allows virtually any food to serve as a vehicle for transmission. However, the most common types of foodborne viral diseases are hepatitis A, infectious hepatitis, and acute viral gastroenteritis. The hepatitis A virus, HAV, is one of more than 70 members of enterovirus group of Picornaviridae family. HAV is presumed to replicate initially in the gastrointestinal tract and then spread primarily to liver, where it infects hepatocytes and Kupffer cells. Food-borne viral gastroenteritis, on the other hand, is usually a mild disease with various degrees of nausea, diarrhea, malaise, abdominal pain, muscle pain, headache, and low-grade fever. Ice, water, ice cream, milk, pastries, salads, sandwiches, shellfish, and other foods consumed raw or subjected to additional handling after cooking are major food vehicles for virus transmission. The highly pathogenic avian influenza H5N1 virus is a fast re-emerging pathogen. It has killed millions of poultry in a number of countries throughout Asia, Europe, and Africa. Although avian influenza viruses are essentially animal diseases, the highly pathogenic H5N1 is able to infect and kill humans. Health experts are concerned that the coexistence of human flu viruses and avian flu viruses, especially H5N1, will provide an opportunity for genetic material to be exchanged between species-specific viruses, possibly creating a new virulent influenza strain that is easily transmissible and lethal to humans. 3.4.4 Diseases by parasites. Numerous parasites can be transmitted by food, including many protozoa and helminths. The most common foodborne parasites are protozoa such as Cryptosporidium spp, Giardia intestinalis, Cyclospora cayetanensis, Toxoplasma gondii, and Entomoeba histolytica. Roundworms such as Trichinella spiralis and Anisakis spp, and tapeworms such as Diphyllobothrium spp and Taenia spp. Many of these organisms can also be transmitted by water, soil, or person-to-person contact. Occasionally in the developed countries, but often in developing countries, a wide variety of helminthic roundworms, tapeworms, and flukes are transmitted in foods such as times undercooked fish, crabs, and mollusks, times undercooked meat, raw aquatic plants such as watercress, times raw vegetables that have been contaminated by human or animal feces, times foods contaminated by food service workers with poor hygiene or working in unsanitary facilities. Symptoms of foodborne parasitic infections vary greatly depending on the type of parasite. Protozoa such as Cryptosporidium spp, Giardia intestinalis, and Cyclospora cayetanensis most commonly cause diarrhea and other gastrointestinal symptoms. Helminthic infections can cause abdominal pain, diarrhea, muscle pain, cough, skin lesions, malnutrition, weight loss, neurological, and many other symptoms depending on the particular organism and burden of infection. Treatment is available for most of the foodborne parasitic organisms. 3.4.5 Diseases by natural toxins. 1. Ciguatera poisoning. 2. Shellfish toxins, PSP, DSP, NSP, ASP. 3. Scombroid poisoning. 4. Pyrrolizidine alkaloids. 5. Phytohemagglutinin, red kidney bean poisoning. 6. Granotoxin, honey intoxication. 7. Gempilotoxin, gastrointestinal illness from consumption of E. coli and all fish. 8. Tetrodotoxin, pufferfish. The most commonly occurring natural toxins are times ciguatera fish poisoning. Ciguatera is a form of human poisoning caused by the consumption of subtropical and tropical marine finfish which have accumulated naturally occurring toxins through their diet. The toxins are known to originate from several dinoflagellate algae species that are common to ciguatera endemic regions in the lower latitudes. Manifestations of ciguatera in humans usually involves a combination of gastrointestinal, neurological, and cardiovascular disorders. Symptoms defined within these general categories vary with the geographic origin of toxic fish. Times shellfish poisoning. Shellfish poisoning is caused by a group of toxins elaborated by planktonic algae, dinoflagellates, Gonulax catenella, and G. tamausis in most cases, upon which the shellfish feed. The toxins are accumulated and sometimes metabolized by the shellfish. The 20 toxins responsible for paralytic shellfish poisonings, PSP, are all derivatives of saxitoxin. Direct shellfish poisoning, DSP, is presumably caused by a group of high molecular weight polyethers, including ocadic acid, the dinophysis toxins, the pectinotoxins, and lysotoxin. Neurotoxic shellfish poisoning, NSP, is a result of exposure to a group of polyethers called brevtoxins. Amnesic shellfish poisoning, ASP, is caused by the unusual amino acid, domoic acid, as a contaminant of shellfish. Types of shellfish poisoning times paralytic shellfish poisoning, PSP, times direct shellfish poisoning, DSP, times neurotoxic shellfish poisoning, NSP, times amnesic shellfish poisoning, ASP, times scombroid poisoning, also called histamine poisoning. Scombroid poisoning is caused by the ingestion of foods that contain high levels of histamine and possibly other vasoactive amines and compounds. Histamine and other amines are formed by the growth of certain bacteria, proteins moldini, Klebsiella pneumoniae, and the subsequent action of their decarboxylase enzymes on histidine and other amino acids in food, either during the production of a product such as Swiss cheese or by spoilage of foods such as fishery products, particularly tuna or mahi-mahi. However, any food that contains the appropriate amino acids and is subjected to certain bacterial contamination and growth may lead to scombroid poisoning when ingested. 3.4.6 Diseases by prions. Prions are normal proteins of animal tissues that can misfold and become infectious. They are not cellular organisms or viruses. Prions are associated with a group of diseases called transmissible spongiform encephalopathies, TSEs. In humans, the illness suspected of being food-borne is variant Creutzfeldt-Jakob disease, VCJD. The human disease VCJD and the cattle disease, bovine spongiform encephalopathy, BSE, also known as mad cow disease, appear to be caused by the same agent. 3.5 Types of food-borne diseases, FBDs. Types of food poisoning include the following: times intoxications, times infections, times toxi-infections. Table 3.2 enlists the food-borne diseases and causative pathogens. 3.5.1 Intoxications. Intoxications involve food poisoning in which the organism grows in the food and releases a toxin from the cells. When the toxin is ingested along with the food, the toxin gives rise to the food poisoning syndrome, signs and symptoms that indicate a particular disease. The presence of the organism in the food is irrelevant to disease production. It is the toxin that gives rise to the disease. Bacterial toxins that produce intoxications are exotoxins that are either enterotoxins affecting the gut, as in the disease caused by Staphylococcus aureus, or neurotoxins, as in the disease caused by Clostridium botulinum, the toxin in this case affecting the nervous system. Mycotoxicosis, diseases produced by the ingestion of food containing mycotoxin, produced by moles, and the diseases produced by algal toxins that find their way into shellfish can also be considered intoxications. Generally, intoxications have short incubation periods, time from ingestion of the food to the appearance of symptoms. 3.5.2 Infections. Infections involve food poisoning caused by the ingestion of live organisms when, typically, the organisms grow in the gastrointestinal tract to produce the disease. Most food poisoning caused by microorganisms falls into this category. For example, food poisoning caused by Salmonella spp, salmonellosis. Enteritis, associated with food poisoning infections, is due to the production of exotoxins or endotoxins that act as enterotoxins. 3.5.3 Toxi-infections. In some types of food poisoning, e.g. Clostridium perfringens, live cells need to be ingested for the disease to occur, but the organism does not grow and reproduce in the gut. Vegetative cells sporulate after ingestion, and an enterotoxin is released when the spore mother cells break down, releasing the toxin. Because living cells also need to be ingested to cause this type of food poisoning, it can be considered as a food-borne toxi-infection. Table 3.2 Microbial food-borne diseases and causative pathogens. Look at the screen. 3.6 Common foodborne pathogens and their symptoms. Symptoms will vary depending on the type of pathogen and may include diarrhea, vomiting, nausea, abdominal pain, and fever. Other symptoms may include headache, jaundice, and numbness. Symptoms can take between a few hours to a few days to develop and may last for a few days, depending on the type of pathogen. Some common pathogens and their features. The foods shown in the table have previously been found to be the source of the pathogens listed. This does not mean that these foods are always unsafe to eat or that such pathogens are always present. However, the foods should be purchased from a reputed supplier and handled with care. Look at the screen. 3.7 Factors responsible for FBD. Several factors have developed during the past 20 years that make the emergence of foodborne diseases a reality. Before discussing these new risk factors, it would be good to review the 5 basic factors that must be present in order for a foodborne disease to occur. When reading about the newly developed factors that predispose society to foodborne diseases, keep these basic factors in mind. 1. Presence of pathogenic organisms or toxins in the food consumed. 2. Source of contamination. 3. Medium for the growth and maintenance of the pathogen or toxin. 4. Proper environmental conditions to maintain or allow for replication of the pathogen. 5. Consumption of a significant quantity of the contaminated or infectious food. I acquire an infectious dose. Table 3.3 Factors contributing to outbreaks of foodborne disease. Various sources. Contributing factors percentage. A. Factors relating to microbial growth. Storage at ambient room temperature 43%. Improper cooling 32%. Preparation too far in advance of serving 41%. Improper warm holding 12%. Use of leftovers 5%. Improper thawing and subsequent storage 4%. Extra large quantities prepared 22%. B. Factors relating to microbial survival. Improper reheating 17%. Inadequate cooking 13%. C. Factors relating to contamination. Food workers 12%. Contaminated processed non-canned foods 19%. Contaminated raw foods 7%. Cross-contamination 11%. Inadequate cleaning of equipment 7%. Unsafe source 5%. Contaminated canned foods 2%. 3.8 Emerging foodborne diseases. The basic definition of an emerging or re-emerging infectious disease is a disease whose incidence has increased in a defined time period and location. If the disease was unknown in the location before, the disease is considered to be emerging. However, if the disease had been present at the location in the past and was considered eradicated or controlled, the disease is considered to be re-emerging. Diseases considered to be emerging or re-emerging include avian influenza, West Nile virus, bovine tuberculosis in wildlife, etc. Recognition of an emerging disease can occur because the disease is present in the population for the first time, because the disease has been detected for the first time, or because links between an infectious agent and a chronic disease or a syndrome have only recently been identified. Many of these emerging diseases are zoonotic and rely on animal populations as reservoirs of infection. Most emerging infections are caused by pathogens already present in the environment, brought out of obscurity or given a selective advantage by changing conditions and afforded an opportunity to infect new host populations. These changes include ecological changes, such as those due to human activities or to anomalies in climate, demographic changes and behavior, travel and commerce, technology and industry, microbial adaptation and change, and breakdown of public health measures. Many factors precipitate emergence by placing humans or animals in contact with a natural reservoir or host for an infection unfamiliar but already present, often a zoonotic or arthropod-borne infection, either by increasing proximity or often also by changing conditions so as to favor an increased population of the microbe or its natural host. New food-borne disease threats occur for a number of reasons. 1. Globalization of the food supply. A large outbreak of cyclosporiasis occurred in North America in 1996 to 97 linked to contaminated raspberries imported from South America. 2. Inadvertent introduction of pathogens into new geographic areas. Vibrio cholerae was introduced into waters off the coast of southern United States when a cargo ship discharged contaminated ballast water in 1991. 3. Travelers, refugees, and immigrants exposed to unfamiliar foodborne hazards while abroad. International travelers may become infected by foodborne pathogens that are uncommon in their countries. It is estimated that about 90% of all cases of salmonellosis in Sweden are imported. 4. Changes in microorganisms. Changes in microbial populations can lead to the evolution of new pathogens, development of new virulent strains in old pathogens, development of antibiotic resistance that might make a disease more difficult to treat, or to changes in the ability to survive in adverse environmental conditions. 5. Change in the human population, the population of highly susceptible persons, expanding worldwide because of aging, malnutrition, HIV infections, and other underlying medical conditions. Age is an important factor in susceptibility to foodborne infections because those at the extremes of age have either not developed or have partially lost protection from infection. Particularly for the elderly, foodborne infections are likely to invade their bloodstream and lead to severe illness with high mortality rates. People with a weakened immune system also become infected with foodborne pathogens at lower doses, which may not produce an adverse reaction in healthier persons. Seriously ill persons, suffering, for example, from cancer or AIDS, are more likely to succumb to infections with Salmonella, Campylobacter, Listeria, Toxoplasma, Cryptosporidium, and other foodborne pathogens. In developing countries, reduced immunity due to poor nutritional status render people, particularly infants and children, more susceptible to foodborne infections. 6. Changes in lifestyle. Greater numbers of people go out and eat meals prepared in restaurants, canteens, fast food outlets, and by street food vendors. In many countries, the boom in food service establishments is not matched by effective food safety education and control. Unhygienic preparation of food provides ample opportunities for contamination, growth, or survival of foodborne pathogens. 3.8.1 Examples of emerging foodborne diseases. Some foodborne diseases are well recognized but are considered emerging because they have recently become more common. For example, outbreaks of salmonellosis have been reported for decades, but within the past 25 years the disease has increased in incidence on many continents. In the Western Hemisphere and in Europe, Salmonella serotype enteritidis, SE, has become the predominant strain. Investigations of SE outbreaks indicate that its emergence is largely related to consumption of poultry or eggs. While cholera has devastated much of Asia and Africa for years, its introduction for the first time in almost a century on the South American continent in 1991 makes it another example of an infectious disease that is both well recognized and emerging. While cholera is often waterborne, many foods also transmit infection. In Latin America, ice and raw or underprocessed seafood are important epidemiological pathways for cholera transmission. Other foodborne pathogens are considered emerging because they are new microorganisms or because the role of food in their transmission has been recognized only recently. Infection with Escherichia coli serotype, O157, H7, E. coli, was first described in 1982. Subsequently, it has emerged rapidly as a major cause of bloody diarrhea and acute renal failure. The infection is sometimes fatal, particularly in children. Outbreaks of infection, generally associated with beef, have been reported in Australia, Canada, Japan, United States, in various European countries, and in southern Africa. Outbreaks have also implicated alfalfa sprouts, unpasteurized fruit juice, lettuce, game meat and cheese curd. In 1996, an outbreak of Escherichia coli O157H7 in Japan affected over 6,300 school children and resulted in 2 deaths. This is the largest outbreak ever recorded for this pathogen. Times Listeria monocytogenes, LM, is considered emerging because the role of food in its transmission has only recently been recognized. In pregnant women, infections with LM can cause abortion and stillbirth, and in infants and persons with a weakened immune system it may lead to septicemia, blood poisoning, and meningitis. The disease is most often associated with consumption of foods such as soft cheese and processed meat products that are kept refrigerated for a long time because LM can grow at low temperatures. Outbreaks of listeriosis have been reported from many countries, including Australia, Switzerland, France, and the United States. 2 recent outbreaks of Listeria monocytogenes in France in 2000 and in the USA in 1999 were caused by contaminated pork tongue and hot dogs respectively. Times foodborne trematodes are also emerging as a serious public health problem, especially in Southeast Asia but also in Latin America, in part due to a combination of increased aquaculture production, often under unsanitary conditions, and of consumption of raw and lightly processed freshwater fish and fishery products. Foodborne trematodes can cause acute liver disease and may lead to liver cancer. An estimated 40 million people worldwide are affected. Times bovine spongiform encephalopathy, BSE, a fatal, transmissible, neurodegenerative disease of cattle, was first discovered in the United Kingdom in 1985. The cause of the disease was traced to an agent related to scrapie in sheep, which contaminated recycled bovine carcasses used to make meat, and bone meal additives for cattle feed. Recycling of the BSE agent led to a distributed common source epidemic of more than 180,000 diseased animals in the UK alone. The agent affects the brain and spinal cord of cattle udder, and lesions are characterized by sponge-like changes visible in a microscope. At this time, 19 countries have reported endemic BSE cases, and the disease is no longer confined to the European community. A case of BSE has been reported in the cattle herd of Japan. In human populations, exposure to the BSE agent, probably in contaminated bovine-based food products, has been strongly linked to the appearance in 1996 of a new transmissible spongiform encephalopathy of humans called variant Creutzfeldt-Jakob disease, VCJD. List of emerging food and waterborne pathogens, bacteria. Times diarrheagenic E. coli, E. coli 0157H7, times pathogenic Vibrio, times Shigella species, times Salmonella serotype enteritidis, say, times Listeria monocytogenes, times Campylobacter jejuni, times Yersinia enterocolitica, viruses, times caliciviruses, times hepatitis A, times chikungunya virus, times severe acute respiratory syndrome associated coronavirus, SARS-CoV, times avian influenza, protozoa, times Cryptosporidium parvum, times Cyclospora cayetanensis, times Giardia lamblia, times Entomoeba histolytica, times Toxoplasma, times Microsporidia. Prions 3.8.2 Antimicrobial resistance and foodborne pathogens. Bacteria become resistant to antibiotics as a consequence of antibiotic use in humans and animals. Antibiotics used in humans can lead to resistant bacteria that can be spread in communities. For example, there are significant increases in resistant pneumococcus, a cause of pneumonia, gonococcus, the cause of gonorrhea, and Mycobacterium tuberculosis, the cause of tuberculosis. In addition, many pathogens that are spread among patients in hospitals are highly resistant, and these antibiotic-resistant infections can be life-threatening and untreatable with currently available antibiotics. The use of antibiotics in animals similarly leads to resistant bacteria in animals. Bacteria from healthy animals can contaminate food and cause human illness when the bacteria from animals are resistant to antibiotics. The resulting human infection may be more difficult to treat. There is a clear relationship between the use of antibiotics in animals and the appearance of resistant human infections. 1. Particular class of antibiotics, fluoroquinolones, has been particularly important in the treatment of severe infections of humans. Resistance to fluoroquinolones has emerged in some human pathogens. Therefore, collaborative effort, designed specifically to detect the emergence of antibiotic resistance in foodborne pathogens, is an important component of the efforts to improve surveillance for new and emerging pathogens. 3.9 Summary: Foodborne disease generally results from consuming food or drink contaminated with pathogenic bacteria, viruses, or toxins. It often produces symptoms such as nausea, vomiting, stomach pain, diarrhea, or fever. The symptoms are often the same as for gastrotype illnesses caused by other sources, other people carrying the bacteria, unhygienic surfaces, etc. This usually makes it hard to separate illness caused by food from the other sources of illness. Bacteria are found in many places, including in human and animal feces. When these potentially harmful pathogens enter the food supply, they can cause foodborne diseases. Foodborne diseases can occur from any fresh or processed foods consumed in a range of settings, such as homes, restaurants, large catering establishments, schools, and institutions. Most cases of foodborne diseases can be avoided through good hygiene and good food handling practices. Most foodborne diseases are caused by pathogenic bacteria or viruses in food. Other less common foodborne diseases occur from accidental chemical poisoning and natural contaminants. The most common types of foodborne diseases are times bacterial, example Salmonella, Campylobacter, E. coli, Shigella and Listeria, times viral norovirus, rotavirus and hepatitis A, times intoxication caused by toxins produced by pathogens such as Staphylococcus aureus, Bacillus cereus and Clostridium perfringens. Symptoms will vary depending on the type of pathogen and may include diarrhea, vomiting, nausea, abdominal pain, and fever. Other symptoms may include headache, jaundice, and numbness. Symptoms can take between a few hours to a few days to develop and may last for a few days, depending on the type of pathogen. New foodborne pathogens like E. coli will continue to be identified, and other known but rare foodborne pathogens like Salmonella enteritidis, may reemerge as important public health problems. Infections may arise from changes in the microbes, changes in the industrial technology that underlies food production and processing, changes in our choices concerning the foods that we eat, how they are prepared, and where we eat them, and changes in the demographics of the population. The increasing number of elderly and chronically ill are at particular risk for severe illness caused by foodborne pathogens. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.